dear students, welcome to biology classes. Myself Divya, lecturer in biology, Shakthi PU College, Shakthi Nagara, Mangalore. In today's session, I will be discussing a chapter called Biological Classification. It is the second chapter in first PUC biology syllabus. In this chapter also, I will be discussing the most highlighting topics, as well as I will also brief you about the important questions which will appear for the examinations. So let us begin. So in this chapter, the important contents go as follows. So as the name is indicating, biological classification. In this, different biologists or different scientists have given different classification systems according to different criteria. The first attempt was done by Aristotle. He classified plants into three separate categories: trees, shrubs and herbs. So mainly what was considered? Morphology was considered. What is morphology, students? Study of external features. So just based on the external appearance, all the plants were divided into three groups, and all the animals were divided into two groups, some having red blood, some without red blood. So this was a simplest system of classification given by Aristotle. Next we have a better classification system which was given by Carolus Linnaeus. He introduced something called two-kingdom classification system. This can be expected as a 1-mark question. Who introduced two-kingdom classification system? A very simple system. All the plants were put into kingdom Plantae and all the animals were grouped under kingdom Animalia. Next we have the most accepted system of classification given by R.H. Whittaker. Make a note of this, very important for 1 mark. Who introduced 5-kingdom classification system? That is R.H. Whittaker. Which are these 5 kingdoms? As it is mentioned, kingdom Monera, to which all the bacteria belong, kingdom Protista, kingdom Fungi, kingdom Plantae and kingdom Animalia. So in the remaining session, I will brief you about different important characters of all these 5 kingdoms. So we shall begin with the first kingdom, kingdom Monera. Kingdom Monera is a kingdom which includes all different varieties of bacteria. Bacteria belong to this particular group. So it is also known as kingdom Prokaryota. This kingdom is called Prokaryota because all the prokaryotes belong to this particular kingdom, which is including bacteria. We have to discuss important features of the groups: Archaebacteria, Eubacteria, Cyanobacteria and a small group called Mycoplasma. So important highlighting features: these are the most primitive organisms. They are existing from a number of years. How they look like? What is their appearance? They are filamentous, like a filament, or some will prefer to be in groups. Then we use the term colonial. Very important feature is there is no true nucleus. What is the meaning of no true nucleus? That is, the nucleus does not have a true nuclear membrane which is covering the genetic material. Hence, bacteria belong to a kingdom called Prokaryota. Important feature of prokaryotes is they don't have a true nucleus, and most of the cell organelles are missing, and cell wall has a component called murin. Other features: the way in which they are getting their food, either by photosynthesis, that is using sunlight and different raw materials, or breakdown of organic compounds, then we use the term chemosynthetic. Majority of them will depend on others for their food. So we use the term heterotrophic. They show a very simple structure. Structurally they are simple, but the functions within the cell is quite complex. This is a unique feature, and very important feature of this group is they show something known as metabolic diversity. As I have stated, there are different ways to obtain food, either by photosynthesis, either by chemosynthesis, or directly depending on other organisms. Hence we use the term metabolic diversity. Diversity means variations. Variations seen in obtaining food or energy. Reproduction can be by asexual methods, by fission, or during unfavorable conditions, spore formation will take place. A very rare method of reproduction is transfer of DNA from one cell to another cell. So that was about important features of the first kingdom, that is Monera. So features of kingdom Monera, what we just discussed now. This can be asked for 5 marks. Make a note of this. List out the important features of kingdom Monera. Next important topic which we need to discuss is name the types of bacteria based on the shape. This is a very important 2-mark question. Most frequently asked in the examinations. List out the various types of bacteria. As it is mentioned on the screen, some of them appear round in shape, spherical, coccus bacteria. Some will appear to be rod shaped, that is bacillus. Some have a comma shape. This is comma shaped bacteria, and some will be spiral in shape, which is called spirillum. These are the different varieties of bacteria based on the shape. So here there are some images to support what I have just said. This is coccus, bacillus, rod shaped bacteria. This is Vibrium, comma shaped, and the last one, that is spirillum. Next we need to highlight the different groups coming under kingdom Monera. Different groups. One of the group is Archaebacteria. This is the most primitive group. During evolution of life, this is one group which were first formed, or they were living from a number of years. That is why it is mentioned representing early forms of life, and the cell wall component is making them have unique features for their survival. Let us see what are the unique features which helps them in surviving. One is they can survive even if oxygen is not there. They can survive increased temperature conditions. Increased salinity also can be tolerated, or increased acidic conditions of the soil can be tolerated. There are different types of Archaebacteria, and it is mainly depending on the tolerance to different conditions. One type is called halophiles. They can tolerate increased salinity conditions. So you will find these bacteria in salty regions. There is a group which can tolerate high temperature as well as increased acidic conditions of the soil. This group is called thermoacidophiles. And one more group is called methanogens. Now why they are called methanogens? These are the bacteria which will produce methane gas. These are mainly found in regions or in soil where there is high organic content, and during the process of decomposition or degradation, they are synthesizing methane gas. You will also find them in ruminant animals. Ruminant animals are the herbivorous animals like cows and buffaloes, which have an additional chamber called rumen in their digestive tract. Hence, they are called ruminants. In the dung of these animals also you will find these organisms, methanogens. Now, whatever is discussed till now, each of this, that is halophiles, thermoacidophiles and methanogens, you can expect it for 1 mark each. Or for a 3 marks question, name the different types of Archaebacteria, highlighting the important features. Now we need to tell or discuss about a very important group. Examination point of view also it's very important. Mycoplasma. These are also bacteria, but they show some unique feature. They are the ones without a cell wall. Cell wall is missing. Hence, they can easily change their shape. And these are said to be the simplest, smallest prokaryotes. The smallest living cells are mycoplasma. So, 2 important features. One is they don't have a cell wall. One more unique feature is they can survive even without oxygen. So these are the 2 important features of a group called mycoplasma. For your exam, you can expect a very important 1 mark question. Name the smallest, simplest living cells, or name the smallest living prokaryotes, and write unique features of mycoplasma for 2 marks. Next, we need to concentrate on a group called cyanobacteria. Now, cyanobacteria are commonly called blue-green algae. Why they are named so? Because of the pigments and which coloration is shown, hence it is named as blue-green algae. They are not true algae, but they are similar to appearance in algae, hence they are called blue-green algae. Now, Where do you find them? Either in marine habitats or terrestrial habitats. They are either living in groups or they are filamentous. They are having a component called chlorophyll A, which is mainly imparting the green color to this particular bacteria. In this way, they are similar to plants. They are photosynthetically autotrophic. And their colony or the filament is generally having a covering of gelatinous sheath. Sheath means covering. And very important point here is, it is forming a huge mass called algal blooms. These bacteria will grow extensively in water bodies to form algal blooms. A very important feature is they are having the ability of fixing nitrogen. Nitrogen fixation is a process in which available nitrogen is trapped and it is converted into nitrates and nitrites. So the cells which are fixing atmospheric nitrogen here are heterocysts. Very important for 1 mark. Please make a note of this. What are heterocysts and what is the function of heterocysts separately can be asked. Examples also can be asked for 1 or 2 marks. Nostoc and Anabina. So in this image, what you can see is there is a water body and this region is completely green in color. This is because of presence of or extensive growth of cyanobacteria which form algal blooms. So what are algal blooms can be an important 1 mark question. Huge masses of cyanobacteria or blue-green algae growing in water bodies. So this is a diagram of Nostoc filament. For your exam, this simple diagram can be asked for 2 marks. 2 mark question, draw a neat label diagram of Nostoc filament. This is how Nostoc looks. All these are the cells which are arranged in a chain-like fashion, vegetative cells which carry out the functions of the entire filament or the colony. There are specialized cells called heterocysts. As I mentioned previously, these are the cells which help in a process called nitrogen fixation. Heterocells help in nitrogen fixation and the filament is having a covering of gelatinous sheath which is also called mucilaginous sheath. So this is important for 2 marks. Make a note of this, students. So this is a colony of Nostoc. Next, very important for 5 marks, list out the features of heterotrophic bacteria. What are heterotrophic bacteria? These are the bacteria which will depend on others for their food. Hetero means different. So what are the important features? You will find them in very huge numbers, most abundant. They are mainly functioning in decomposition, that is saprophytic. Whatever organic waste is there, it is converted into smaller organic content with the help of bacteria. The group is heterotrophic bacteria. Certain examples, there are some bacteria which also give us some uses or benefits. Simplest one is lactobacillus, which is used for converting milk into curds. Then certain bacteria can be used in synthesis of antibiotics. Just now I had just discussed, some of them help in nitrogen fixation. Majority of them cause diseases. A number of diseases are caused by bacteria. You just need to know the names. Cholera, tetanus, typhoid, plague and pneumonia are different bacterial diseases. So we have finished discussing kingdom Monera. Under this, one is features of the kingdom you need to remember. Then classification with various examples. Next we are going to the second kingdom, that is kingdom Protista. Here you can expect an important 2 mark question. Write down the important groups coming under kingdom Protista. Which are these groups? Chrysophytes, dinoflagellates, euglenoids, slime molds and protozoans. So just the names of different groups can be asked. So important highlighting features of each group. Chrysophytes, under this there are 2 important members, diatoms and golden algae. So in this image, diatoms have been enlarged and shown. The main cell wall composition of diatoms is silica. Because of this, though the organism dies, the cell wall material will remain for a number of years. So what is diatomaceous earth? Accumulation of cell wall deposits of diatoms which is having silica in the cell wall for a number of years. Make a note of this, students. This is very important for 1 mark. Define diatomaceous earth, or what is diatomaceous earth. Second group is dinoflagellates. What is the meaning of this? Di means 2. Flagellates is referring to flagella. So this is a group which has the presence of 2 flagella. Very important under this, among different types, are red dinoflagellates which appear red. They multiply very fast, converting a part of the water body to appear red in color. This is what is called red tides. A good example for a red dinoflagellate is goniolax. This will release toxic chemicals which can kill aquatic animals like fishes. So here, examination point of view, one example is, or for 1 mark, give an example for a red dinoflagellate, very important. And for 1 mark, you can also expect what are red tides. So this is an image of a dinoflagellate, and this is an image showing red tides. As you can see here, an entire huge area has been completely converted to appear red because of red dinoflagellates. Next important group is euglenoids. This is a member, or this is a group which instead of cell wall they have a flexible layer called pellicle. This can be asked for 1 mark. So this is an example which comes under euglenoids, euglena. It also has flagella. Next group is slime molds. So here important feature is they carry out decomposition, saprophytic, and during certain conditions they will form aggregations called plasmodium, which will later differentiate into reproductive structures called fruiting bodies. Fruiting bodies later will produce spores. So this plasmodium can be asked for 1 mark. Plasmodium are aggregations of slime molds produced during certain conditions. So here this is an image of a slime mold which has formed a huge mass, as you can see, and it is named as plasmodium. Last group is protozoans. Examination point of view, very important. What is asked? Name the different types of protozoans, highlighting one important feature and giving example for 3 marks, or sometimes they also ask for 5 marks. So which are these different groups? Amoeboid, flagellated, ciliated and sporozoans. Very important. Now, one very important feature of each of the group is there is a particular type of locomotory structure. What is locomotion? Ability of the organisms to move. And the structures helping are called locomotory structures. So in this particular group, they have locomotory structures called pseudopodia. Now if I draw amoeba, this is how we will draw, isn't it? Irregular in shape, having a nucleus. What are these extensions? These extensions are nothing but pseudopodia. Okay, so this is a unique feature of amoeboid protozoans. Good example is, one is amoeba. The one which is causing disease is entamoeba, parasitic. So this is image of amoeba. All these projections, what is highlighted is pseudopodia. This image is entamoeba, the one which causes a disease called amoebiasis. Next group is flagellated. The name itself is telling us this is a group which have flagella as their locomotory structure. Good example is trypanosoma. This causes a disease called sleeping sickness. Why it is called sleeping sickness? The person will have problems related to sleep. So this is very important for 1 mark, students. Name the organism, name the protozoan which causes sleeping sickness. This is an enlarged view of one trypanosoma organism. Next group is ciliated. As the name is indicating, the locomotory structures are a number of cilia. Good example is paramecium. So this is image of paramecium. Here throughout the extensions, fine extensions which are hair-like are called cilia. Last group coming under protozoans is sporozoans. Now this is a group which mainly live as parasites. What is the meaning of parasite? A parasite is an organism which requires another organism to survive and to reproduce. The other organism which it is using is a host. So sporozoans mainly cause diseases. They are parasitic. A good example for a protozoan which is a sporozoan is Plasmodium. Plasmodium causes malaria. And the vector which is transmitting malaria is female Anopheles mosquito. Female Anopheles mosquito is a vector, that is the carrier of the disease. The main causative organism is Plasmodium. So here one more question can be asked. Name the protozoan which is causing a disease called malaria, or they can twist and ask, or they can ask an indirect question, which disease is caused by Plasmodium, that is malaria. Dear students, in this session, I have briefed the important topics coming under biological classification, in which I have highlighted different classification systems first, given by different biologists, and later I discussed important features of two kingdoms. One is kingdom Monera and then kingdom Protista. I hope you have understood all the topics what is taught in this session. I will see you in another session in a different video. Thank you, students.